The Portent Tide Research Podcast is about interviewing researchers and providing an insight into their field and how they think of their field overall. It is specific in exploring and documenting some of their current or past research, from the preconception of their hypothesis right through to the communication of their findings, delving into their decision-making process along the way. And the discussion is generalized in hearing their thoughts on key themes within research and what their line of work takes and requires of them. The aim of Portent Tide is to put the science researchers undertake into a format that is more engaging, with the capacity to reach further out to larger audiences, with a determination to maintain quality and trust with researchers by publishing the discussion in full only upon the approval of the researcher. This podcast is a conversation with Efrain Escudero and Jeffrey Lackmann. Both are exploring research in relation to fungi, and more specifically, the relationship between fungi and coffee plants. Efrain is completing his PhD studying the effects of fungi within coffee plantations. He explains that at any given time within the lab he is part of, there are multiple ongoing hypotheses, and explains how these questions consistently arise due to the part of the mycology field he is part of being relatively young. One such hypothesis Efrain mentions is the exploration of how the relationship between fungi and coffee plants interact with pathogens and parasites. Jeffrey is completing his master's degree and was at the biological station working in the capacity as a researcher, looking into fungi growth within the coffee plant leaf. These fungi are known as endophytes. Coming into the Las Cruces Biological Station, I was unaware of exactly what research was being conducted, so upon meeting and spending time with Efrain and Jeffrey, I asked if we could start the conversation by having them explain how they built their knowledge of fungi, with an intent to provide a potential novice of the kingdom of fungi with a path to being a professional mycologist if someone wishes to do so. As you can see, I make a fool of myself at times using the incorrect terminology, but I'd hope in some way this reflects that complex subject matter is accessible provided the right questions are asked. Finally, this conversation was a great insight into how to navigate key themes of research, and a window into how asking these questions can generate different viewpoints and strategies for long-term success and health in research as a career. Thank you for listening and enjoy the podcast.
2 minutes.
Right. Yeah.
Sweet. Well, again, I say this to everyone, thank you for sitting down with me, and it's just it's a pleasure to speak with you guys. So introduce yourselves and professional backgrounds. Or
Sure.
education background
Sure.
as well, yeah.
Yeah. So my name's Jeffrey Lackmann. I'm a master's student for NDSU, North Dakota State, and I got my undergrad in English from Luther College in Iowa, and so I'm finishing up my first year master's program, completing most of my field work for my thesis right now. Yeah.
Awesome. Efrain?
Well, my name is Efrain Escudero, so I'm a biologist. I studied in Mexico, the bachelor degree and my master's degree also. I've been working with fungi maybe for 7 years now. This is my last year of the PhD program. So I'm basically studying endophytic fungi that could be useful as biocontrollers for solving coffee diseases. Mm-hmm.
Awesome. Cool. So I think we talked about starting today was just a bit differently, we're starting today is a bit differently to how I normally do it, just because I think it'd be cool to, as I said, to purely explore a lot of fungi, and especially because I don't have a background in it, and I think it is a different world. So I thought, that question that we could start with was the one I gave you, Jeffrey, about where you started when you first started learning about fungi, and what was the process they introduced each new topic, in what sequence,
Mm-hmm.
And then when we wrap that kind of topic up, I guess how you would do it yourself, how you would structure a course yourself if you were to try to teach some new people.
Mm-hmm.
And maybe we can compare both of your education, because when did you start learning about fungi, Efrain?
I think My first approach with fungi, it was on 2013 maybe. Yeah, I was in a project that consists basically in identifying all the edible fungi in markets in Oaxaca, in Mexico. So it is known that, for example, Mexicans are like a mycophile society. We eat fungi for tradition, and in some markets it is incredible the variety that you can find. So we were trying to see the species and also if they were selling them as edible only or sometimes medicinal. So that was my first approach to mycology.
Okay, so it was more informal and
Informal.
and just going to the market.
Yeah.
How is this? Was this like a once a week, like a weekend type market?
Yeah,
or-
during it was on Saturdays the market.
Okay. And it was
Yeah.
quite popular.
Yeah, it's popular for the region because it's like a particular part in the state of Oaxaca, which is Tlaxiaco. So it's on the like in the middle something in the street of the state. So what we were basically doing was ethnomycology, which is the study of fungi and the relationship with humans. So.
Okay. All right. Sorry, guys. Just let me do one thing with this camera over here. But yeah, Jeffrey, start by.
Yeah,
go ahead.
yeah. So, so I, like I said before, I graduated with a degree in English, so I didn't have a ton of experience with fungi. But after I graduated, I got really interested in foraging in the woods around Minnesota, and also really interested in nutrient cycling, so how things move through a forest and or prairie. And so a lot of my education was really just sort of self-directed. I would read things on my own and try to figure out kind of the life cycle I started with Basidiomycetes and then branched out from there. But when I went to graduate school, I took a course, fungal biology, that gave me sort of the foundation for what I do now. And the approach for that course was the first few units are just very basic sort of fungal anatomy. So what's the difference between the different fruiting structures, and what's the difference between different hyphae and cenocytic hyphae versus other kinds of hyphae. And so then once you have that foundation, we just sort of went through the larger group. So Basidiomycetes, and then Ascomycetes, and then we finished out basically going group by group, and then with slime molds and then aquatic fungi. And yeah.
Can you explain the word hyphae?
Yeah. So a hyphae is like an arm of mycelia is this collection of hyphae, and it's the body. It's a single cell that extends and feeds. So the tip of the hyphae, the hyphal tip, is what's actually releasing and growing, and it exudes it exudes metabolites to break down the environment, and then it can absorb the nutrients that way.
Okay. So I kind of brought up that topic about how the fungi sense their environment. Probably not the right word, using sense, but so that seems to go into that sort of territory about how it's gaining information or giving out information.
Right, yeah. So, like, for example, there's studies that show how branching happens. So, like, if a hyphae's growing and it's growing singly, and then you stress it by putting a plate on top, it will grow. It will diverge.
Right.
And so, its branching pattern is sometimes a result of sort of stimuli. So, if it finds a lot of nutrients here, it's going to pull resources, and hyphae will converge on a nutrient-rich area. And if it needs to explore because there's not enough nutrients, it will explore in different ways. Yeah.
Okay. And it's just sort of, while it's already, say it has a good source of whatever it's gaining, is it still exploring at the same time? Is it just focusing on this one source and just putting all resources towards that, or is it trying to explore different avenues at the same time?
I think in large part that may depend on conditions in the species, but just in my own observations, yeah, there's some they're not just going in this direction. There's some exploration happening, because in a given environment, it's beneficial to be, if there's two food sources and you can get to both, it'd be beneficial to have both. Yeah.
Sure. So what are your thoughts, Efrain, on whether is it good to start with anatomy first, or could be good to
with
do
the taxonomy? Yeah, it's like the basic, and it's the basis for understanding all the major groups, and with that you can move forward to what they do on nature. So you can classify them on different ecological guilds, and you will see that there's a variety of functions for them. Some are not restricted to one guild, for example. They can have two or maybe three types of guilds. So for example, you can see that maybe a fungi is a parasite, maybe with the other life cycles, because they have two life cycles. Then it's not a parasite, maybe it's a saprotroph. So that's interesting because they are like very well suited to adapt to almost any kind of environment. So to realize how they do it, you need like a stronger basis on taxonomy to recognize the structures and what they do.
So within fungi education, you're really saying that the anatomy is the foundation. Because I guess I asked the question because sometimes you can approach learning about species from a population level or more of a landscape level. So I was wondering whether or not there is that sort of approach that's less specific but more generalized.
I mean, I guess the way that we started the class, it was like half of a lecture, but basically we split it into sort of this friend or foe type thing. So are fungi good for people or bad for people? And so then we went through several ways in which they're good. So, you have cheese, and you have alcohol, and you have medicine, and that sort of thing. And then bad, you have plant pathogens, rusts that devastate crops. And so on a very broad sense, just looking at the application to people's lives and whether or not you need to think of them as good or bad is a good way to get you to start thinking about function. Because if you think about, what is a thing doing? Is it helping me? Is it assisting me in making a food, or is it actually stealing my food, ruining my food?
Yeah. Okay, cool. Cool. Let's explore evolution of fungi then. And also evolution of use. So where would you guys start if you were to pick a point there?
Evolution of use.
I guess evolution to begin with, but then also when you're aware of people, and situations you're aware of people, say, starting to explore that are really bedded in evidence.
I think ancient cultures know a lot of fungi. They can recognize like edible and medicinal and also psychotropic fungi. So I think the approach of the humankind with fungi, I think it's very, very old. So it is supposed that it triggers the communication among people, and also some people say that they also are related to the expansion of consciousness. So for example, you can see it in some cultures in Mexico, that they can give a precise name to different species. And if you look at the taxonomy, like the Occidental taxonomy, it's quite similar. And sometimes it resolves better. So that gives you an idea of how old is our relationship with fungi. So, for example, with that's what with fungi that we can see in the forest, the macrofungi, but the things changes with iphomycetes, with the things that we cannot see unless we have a petri dish with agar, for example. hmm So in that way, yeah, we can see the relationship in products, as Jeffrey said. So, but we were not able a few years ago to know that they were fungi. We just knew the process, but we don't know what was happening and then who was acting.
Was there, you were talking about Mexico understanding of the macrofungi. Was there any seemingly understanding of the micro stuff you're talking about? That like there was a use of, say, particular fruits or particular fermentation practices that created results that seemed similar to the macrofungi. Was there any history or something like that?
I think something is known about, for example, tepache, which is like a fermentation of pineapple, and also I think they can use some apples also. So they know that something was acting there, but they cannot realize that it was like a fungi or something. But in the forest, if they see like a log, a dead log with white mycelium, they can recognize that that's the fungi.
Okay. So is how similar are different species of fungi? And I guess I'm asking what other manners you might use to differentiate cousins. Is it something like shape, color, size?
Well, among fungi, I think are one of the most cryptic organisms in the world. So the to be cryptic, it means like look the same in color, shape, size, smell, even, but the changes are on the genomic level. So maybe with fungi uh, will be possible to support the idea to stop talking about species and talk about genomes, for example,
Okay.
which is the new era. So
Right.
this is what is coming. But in the field, that's one of the main reasons it is so difficult to do ecology with fungi, because you can't recognize one individual from another one. So maybe you can talk about maybe populations if you are in a very long distance, but I don't know, they are like super organisms, so
Sure.
it's
So
hard.
can you elaborate on the thing we discussed earlier about the complexity of not recognizing an individual or not recognizing or recognizing populations instead? Just how you might, like, why is it that you can't recognize one? Say, my probable personal opinion, I would have thought I see a fruit and would have thought that was one individual, and then the ones next to it were different individuals, but you're telling me that they're all the same thing. So can we explore? Um, yeah, can we just explore what that kind of idea in general? Why is it that they form such large connections? And maybe the question isn't entirely clear, but yeah, can you
Yeah.
build
But
off
the
that?
thing is that the what we see in the forest is are basically fruiting bodies. So the true fungi is the mycelium, which lies underneath all those fruiting bodies. So you can follow the network if you want to, and maybe the size is of meters. And I can't remember the name of the fungi that lives in the, I think it's in Canada or the US.
Isn't it? Yeah, it's a honey mushroom. Is the fruiting body, and it's the largest organism on the planet, and it's like 50 kilometers wide, perhaps more. I don't know exactly, but it's in the Pacific Northwest. I think in Oregon, or I'm not exactly sure. I don't want to,
Right.
but it yeah.
And so I imagine that they've just taken samples from different locations and tested them and just.
And yeah,
Like,
they're genetically the same
Oh.
organism, individual.
I have to imagine that kind of shook the sort of fungi world. Fungi researchers would have gone
Yeah.
crazy over that, right? Breaking into that, where have there been breakthroughs in the research, in your careers or in the history you're aware of?
Definitely the molecular techniques that we are using to identify fungi are one of the most powerful tools ever made. So all the texts that were written before that era uh, are suffering from big changes. Actually, the big databases that contain the fungal names are changing every 6 months now. So yeah, what is something today, maybe it's something else tomorrow, and some things they didn't have higher labels in the taxonomy. For example, they can they can have like a genus, but they don't know how to put that genus. So now it says Incertae sedis, which is like the family is unknown. So
Okay. And so, what is it that's really changing every 6 months to bring about?
because all all of the taxonomic work that it is done around the world, so there are like specialists on each group. So for example, my advisor works with Trichoderma, which are Hypocralian species, which is the order. So um, before her PhD, I think there were like 20, nearly 20 species of Trichoderma known in the entire world. After her work was done, the count went up to 200 something, and now we have 250 more or less Trichoderma species only. So uh, that's with that case, and those repeats with other groups of fungi. So it is incredible.
Is there projections of where that particular one might expand towards?
Yeah, the the people says that fungi might be the most diverse organisms in the world, more than insects.
Right. Okay, that's
So,
massive.
mhm.
Um, okay. Yeah. So we touched on that uh, radiocarbon. Can you guys explain what was done in that experiment, and what is that? Do you think that is one of the most significant discoveries within the world of fungi? Because I'm only basing that because I'd previously heard about it. But I wonder if that was one of the most significant discoveries, or if there was others.
Well, I mean, it's a very very useful tool to see how nutrients are moving around. So and what things are doing. So if you have say like a plant fungal partnership, and you can create a traceable isotope, and you know where you put it, and then you can see where it ends up. You can say, okay, that is giving carbon to that, not the other way around.
Right.
And you can do that with any number of relationships. So it's a way to sort of make the invisible visible. And there's other more dramatic ways that you can do it too. I mean, you can actually make things phosphoresce. So, like you can under ultraviolet light. So one of the ways people use that is with insects. So if you want to see where an insect moves over a given period of time, you coat it in this substance, and then you run a light, and you can see everywhere it's gone, and you can. So basically the ability to trace where things go in any field is going to be really useful. So like for example with sewage, a completely different example, but if you can dye the water and see where it ends up, you know a lot about how it moves.
Sure.
The same is true for fungi, and if you have the capability to do that, it makes the questions you can ask really widens the scope. Yeah.
Okay. Cool. So, what type of questions have widened that scope that have come from, say, that capacity to map distribution?
Yeah. So, I'm pretty new to the field. I've I haven't read as widely as some, but like some of the things that really have caught my attention are things like tracking where mycorrhizal fungi are moving nutrients in when they're partnering with many different plants. And so it's kind of a pop culture thing now, but thinking about forests as networks and sort of the ability to move nutrients from one place to another, from one species to another, and thinking of fungi as the facilitators of that. I think for a long time people have a plant-centric view, so they see a plant and think of the plant as partnering with the fungi, but perhaps it's clearer to see the relationship as the other way around. The fungi are partnering, are sort of the main driver of the relationship with the plant.
Cool. And let's break into that. We talked on two topics there, with like the market-like structure you introduced, and there was another one. You were discussing that it wasn't always mutualistic, which is perfect what you were saying then.
Yeah. So I think what's really tempting, and because ecology is so complicated and so contingent on so many different factors, that to like define a relationship and say, okay, this is a mutualism, this is a parasitism, this is a commensalism, but we're beginning. I mean, it's intuitive, but starting to see that it's not always mutualistic. It's only mutualistic under certain circumstances. So an example might be like you have a plant and a fungi that are partnering, and during times of plenty, in terms when there's a lot of water in the system, both benefit from the relationship. But when there's not a lot of water, when it's drought, the plant may suffer. The plant may get less resources from the fungi, or the other way around. And so it's not necessarily always clear what is a mutualism and what is not, and whether or not it's always that way.
Okay. Are there many people tempted to paint it that way, like sort of black and white within the field? Is it something that's commonly done, or is it something that's externally done, maybe more media or more pop culture?
Maybe Efrain can speak to this, but I think maybe it's a product of my thinking is that part of it might be a product of textbook learning. So you want to teach a relationship to say this is a mutualism, and show, and then to add a layer of complexity that, okay, well, it's not always a mutualism, is challenging to teach and conceptually. So I think that might have to do with it. But I think generally people are open to, in the field, are open to like contingencies like that. So, but it's very human to just want it to be a simple, you always know how it's working.
Right. Yeah. That makes sense. So that leads into that times of scarcity. Is that when the market-like structure within the mycelium is most observed, or is it also in times of plenty?
Maybe places of plenty is a better thought. In terms of how the study that I'm thinking of. So you have a mycelial, you have an individual that has partnering with multiple plants, maybe over 10 meters, say, and in this part of the plot, there's a lot of phosphorus, and in this part of the plot, there's not a lot of phosphorus. So in the relationship, the fungi is able to basically get the most bang for its buck in terms of what carbon it gives the plant. So the relationship is the plant, or what phosphorus it gives the plant. The plant gives the fungus carbon in exchange for phosphorus. And so in a place where there's a lot of phosphorus in the soil, the plant can actually get some of its own. It doesn't need to rely so heavily on the fungus. And so but in places where phosphorus is low, the plant is kind of desperate. It needs help. And so there are experiments that have shown that fungi can use that to their advantage. They can move carbon over to this low phosphorus area. So using carbon tracing, so like a carbon isotope that we can trace, it starts over here in the high phosphorus, the high phosphorus area, and it moves over here to the low phosphorus area, and then it can exchange that for more money or for more carbon than it would get over here. So it can actually play the market. Yeah.
That's pretty fucking cool. Okay. And where does that information take you in thinking about, say we're studying here in this location. How does that information map out to different biomes? Is that a fair question? Is it do you think it's different?
It's a open, I think, largely open question.
Okay.
So sometimes the resource of scarcity is different. It's not phosphorus. Maybe it's water. Maybe it's communication. So if to know where pests are at. So if something can control that or can manipulate it, that's not the plant. Then I think when you look at a biome, maybe the first step would be to try to determine what is that. There's probably many, but what is the resource that is being exchanged, and is it variable, or is it, because some places have totally universal nutritive makeups and some places are highly variable, so you just have to think about the sort of the what's growing and why, that sort of thing, but yeah.
Okay. Build from there.
Yeah, maybe in that point, you can also see that there are studies that talk about ecological or trophical shifts. So, for example, now we know that some species that now are like mutualistic. Before many years ago, before they were like parasitic. So, a fungus can shift its way of nutrient of how they feed. Depending on the opportunities of the environment. And so, for example, they have improved like cicads. Uh, there are fungi that parasite cicads. So, because cicads are very close to some plant roots, then fungi might change to no, we are not parasiting cicads anymore. So we are going to parasite or maybe live inside the plant. So they become endophytic now, but before they were like a parasite.
And you talk about the same species there.
Yeah, so now they have evolved to be endophytic. So that tells you that maybe in 100 years, maybe they will shift again if this endophytic association is not working for them. They can shift.
Okay, and so we're talking about you're just saying that's over 100 years that
Yeah,
shift.
it's for saying something, but maybe it's 1,000, maybe
Okay.
millions.
Okay, all right.
Yeah, you can date it because there are some techniques that are called like a molecular. You create like a phylogenetic tree, but you add a molecular clock, so you can trace approximately how many years ago this shift was done. So yeah, the scale is large, but.
Okay, interesting. Oh, sorry. Yeah. So the question with the one area of fungi behaving differently in different biomes we previously discussed was dormancy. Could we just quickly cover what that was, and then we'll just explain, like, because you guys spoke about natural disaster, so I'm kind of interested in where that goes. But can you explain dormancy, because you were just talking about it in North Dakota.
Yeah. So in North Dakota, much of the year is very, very cold, and the ground is frozen. So if you have a plant pathogen like a rust, it can rusts have pretty, like wheat stem rust has a very complex life cycle, and so in the summertime it needs to be able to infect plants, but in the wintertime it has to be able to survive to the next year. And so they have developed a life stage of a spore that is melanized, so it's very dark, and it's protected from like UV radiation and freezing temperatures, and it can survive in the soil through the winter. And so this can be really problematic for farmers and agriculturalists, because you have wheat stem rust, but you may have had it from 2-3 years ago, and you've rotated your crops, and it wasn't really a problem with your beets, but then you switch back to wheat and you find, oh, the rust is still there and it's still infecting my plants, and so
Right.
you have to, yeah.
Okay. There's two ways how we can go from that. I was thinking one is we can just break into that agriculture pest versus beneficial, I suppose, but I was also thinking about going towards the natural disasters. So let's go to agriculture first. If we discuss, well, you guys had spoken about okay, how quickly they can shift from a classification of parasitic to
Endophytic.
endophytic, yeah.
Mhm.
Okay, so can we discuss how that might have changed in the world of agricultural farming?
Yeah. mm, in coffee, for example, it is quite interesting that if you isolate fungi from maybe branches and leaves, you will have a lot of Coleotrochum. Uh, and now we know that Coleotrochum has many species. And if you sample some Rubiaceae, which is the Botanica family for coffee, you will also have them. But uh, plants in the forests are not sick. Why the coffee is it? So are the same species? Maybe, but what else? So the rest of the coffee community is the key to understand how is the balance of species present in coffee, which is subjected to a strict management, and with the wild species that are out there with a lot of more beneficial species inside them. So um, that's one of the points. This has been seen in Hevea, which is another tree in the tropics, and also in cacao. So wild cacaos, wild Theobroma, which is the genus, uh, also have more diversity inside them than those Theobromas that are in in agricultural management. So part of this is the surrounding community of plants that can provide like a source of potentially helpful species for those for the coffee, for the Theobroma, for the Bea, whatever. So they give them the balance. It's like with us, with the microbes that we have on our guts. So it's uh, we can shape the community uh, with the things that we eat. So that's why it is recommended to eat fiber and maybe drink a lot of water and stuff like that. So it's basically the same with the plants. So the same happens when we take like antibiotics. We are killing our microbiome. So if you put some fungicides on the plants, you are killing both problematic species, but also maybe you are taking away the helpful species that could be living inside the plant. So now that's why mm, with my study we want to kind of restore the balance in the coffee plants so they can protect itself.
Okay.
Hmm.
Okay. Cool. Um, we'll break into the study in a moment. Just explore. Well, that you just mentioned like a sort of man-made disaster in terms of herbicides or pesticides. How, what what are ways natural disasters can really impact agriculture, but also um, more wild locations as well, and fungal communities?
Uh, the effects are really, you can see it really fast. For example, with hurricanes here in Central America, when a hurricane hits the forest, then you have a lot of trees that are down. So that shapes the community of fungi because you start to, they start appearing like silaroids, which are black fungi. And if the population of them like is like abundant, they can parasite maybe healthy plants. So uh, from a spot of fallen down trees that is like, let's say, like a kilometer, a square kilometer of diameter, then it can become like more, like 2 kilometers. So the impact is huge. So I think there's only a study of how of how hurricanes are impacting the forests in Puerto Rico. So they have they have been able to track the forest recovery and what is happening with fungi. So are they helping the forest to decompose, but also are they helping the forest to recover from the hurricane impact? So the study still is going until now. So it will be interesting to read discussions on it.
Does that break into the idea of what we were talking about? We're talking about historically, what was the word you used? Historically contingent. So in other words, if that hurricane decimates an area, spores are blown into that area, and they're I guess what would be the word, pioneering. Would that be a case?
It could be, yeah, it could be. One of the ways that, so historical contingency is this idea that how a community is shaped depends on what arrives in what order. So, I mean, the easiest way to think about it is like an island, a completely bare desert island, and then you have a species come that is problematic, or that if you have several species arrive, it depends on what order they arrive in. And so, yeah, I mean, natural disaster could essentially create a kind of island within a larger ecosystem, and then what arrives first may impact what community forms. Yeah, for sure.
Okay. Let's break into the studies. Who wants to go first? You want to go first? Okay. So the first question I really have is, prior to beginning, if you imagine that research as a puzzle, how many of the puzzle pieces were already laid out, and in choosing that gap in information, just kind of paint the picture of what you found prior to even having your hypothesis.
So regarding as a puzzle, is interesting because you can see that maybe it's not that there is a huge gap, but small gaps. So you can try to fill that small gaps in order to aid to fulfill a big piece of the puzzle. So I think
Okay, just if you could lean into the mic.
Okay.
Just because the rain is
Yeah.
just now.
Okay, so I think what we do on the lab is basically answering small questions that eventually will answer like big things. So when are we going to have the big thing solved? I don't know. Maybe it will be some, maybe 3 generations after me. So I don't know. So but that's the interesting in science, that you are putting like little stones, and eventually you will have a full brick. And and now choosing which stone you want to support, it's another question, because it depends on what your attention is attracted, what you feel passion for it. So I think a good thing to know that if you are on the right way, it's like despite you are like after a day on the field work on the field, you can still working through the night. So yeah, you are in the right way. You have the passion to keep going. So you will have a lot of challenges during the process of research of any kind. Like I mean, it can be strictly academic, but also like personal, and you will need to take all in balance. So to keep moving forward.
So you feel as though there's just a different set of inputs in that are coming in when you're actually trying to judge what the best thing to study is. So you basically suggested that there was an aim of that lab. What would be, if you, is there like a mission statement of the lab, or is it more of a, or just a generalized, just a,
We can call it like a line, but it's not like a straight line because it's more like a tree because uh, in the lab they are forming students on different fields. Okay, the main issue is fungi, but you can have people working with entomopathogenic fungi. You can have people working with maybe mycorrhizal fungi, uh, and so on. So that's the interesting thing because for answering your question, you will need not only a guide, you will need several more. So the team just grows. And for example, in my case, I have like my principal advisor. She's like, okay, so I'm the expert on fungi, on maybe taxonomy, ecology, and all that stuff. But I can't help you a lot with biocontrol. Okay, so then we need someone who is an expert on biocontrol. And then if I want to answer another question, for example, chemical questions, then I need a chemist in my team. So I'm like the sum of the part of all the advisors, of all the people who's helping me to answer the questions I want.
okay.
So yeah, it's nice.
Okay. Okay. Cool. Yeah. So let's lead into what the hypothesis was, and I suppose more or less how you worded it, but also what variables you wanted to address within that.
So first that we now know that yeah, water reactions are full of uh, most diverse diversity of fungi than the coffee. Second, now we know that there are hypocorallian species that can be useful for the coffee. And now we are on the state on the stage where we are trying a couple of those fungi to see the beneficial effects on the plants. So we wanted to see, for example, a protection effect for against some pathogens, for example, Mycena aceticol or the coffee rust. But now we have some results that says that one of our species is helping with the growth promotion of the coffee plants. So we weren't expecting that, but it's what it came, and so it's good.
Awesome. Okay. Let's break into the method and how you've gone about collecting, deciding to collect results before you arrived here.
Well, I think the methodology always change because you can have an idea that looks perfect and solid, and once you're in the field, it results that no, it is not perfect. I think it is maybe it takes a lot of time to do it. You are run out of resources, so you need to be flexible to make decisions that can be like the right decisions to get the amount of data that is just enough to answer your questions or to keep the work going. So
Maybe break down what are some key factors that you expected and found, expected within the method and found, and expected but you were surprised about. So what were the key factors within the field that you saw were different?
so for example, in the part of the isolation of the fungi, we didn't know what to expect, what the amount of fungi. So we were traveling with a certain amount of petri dishes with agar, and in a field trip, we just ran out of plates. So we need to stop the field trip right there because we didn't have the material to process all the samples. So it was like, I don't know how many petri dishes, 1,300 maybe, something like that. Yeah, and we needed more. So
Okay. And what was the timeline on actually progressing there?
yeah,
Getting those dishes?
we were expecting to process all the samples in a year, but I mean, from the isolation, purification, and identification. So, yeah, I think we were like on time. Uh, and then the thing with that has been even more challenging is like how do I apply this species to the coffee plants? We have tried several kinds of substrates. We were using rice, for example, but the rice get contaminated by yeast really easily. So we changed it to, we tried to use I think horse, was meat for horse, which is made of grass and all kinds of herbs, and it didn't work. So we came back to potato dextrose agar, but in more quantities, and that's how we did it. So
Yeah.
yeah, you need to try several several things until you find the right one. So uh, you lost a lot of time on the way, but maybe it is not lost. It is like it is a time of proving, but you need to hurry up because we all have a deadline,
Sure.
especially for a scholarship. If you have a scholarship or fundings or whatever.
Okay. And so where are you at in this process now? Would you say you're still in the data collection and sort of, it sounds like you're straddling sort of the data collection and results and discussion at the same time.
Yeah, because well, I'm still on the data collection, but on another step. Now I'm collecting. Well, I already have collected chemical data, so that came from my isolates from 4 years ago.
Yeah.
So it is a long process, and it's like a complete different thing of what I've done so far. It's I'm naive. I was naive on the field, so it was challenging, but it was also really cool because you're still learning. So you never stop learning the learning process, and the techniques and the tools that are available now will change tomorrow. So you need to keep going. So not to be stuck in just a way. So yeah, it has been an amazing journey, I think. Yeah.
Sounds let's break into that question that was troubling you for a second. The depicting the results. You were kind of talking about how you were conceptualizing this in your head. So where are you at with thinking about that now? For the right
Yeah.
question.
I think the more results you have and the more things you read, you realize the more or less you know.
Yeah, right.
So it looks like ambitious to try to solve like big questions on any field, not only fungi, but you need to start somewhere. So I think like that now that I have like a vision of how big can a fungus, okay, of how big can a question be related to fungi? Wow, it can have several ways. For example, on the chemistry part, you can be looking for diversity of compounds. But then, if something is interesting, you can keep going only on one compound, for example. And if it results that, okay, it's useful for us or as an antibiotic, then you need other people to get involved to synthesize that compound, for example. So, yeah, it's crazy. So, how far I want to take my results? Well, I need to remind myself that I I decided to do some things with only on the level of describing what is that every fungus have. So that's it. I can keep working on the next steps, but I need like finish that race today. So
Yeah.
okay with that one.
So the complexity seems that you, as you were saying to me earlier, that you change the depiction, you're changing what you're trying to show about this, and so is that sort of what you're battling with there? There's just like the changing that depiction, or how you present the results ultimately changes what you're saying about this.
Yeah.
Is
Yeah,
that a fair way to?
because the results can be like diverse also. So you can tell a big story with your results, but you can split it in like small stories. So on what depends that? It depends on the way you want to write it, because sometimes like a large article can be like boring, but if you split it in small stories, then you can sell it better. So, and it's more like attractive for the public.
I see what you're saying. So actually, even at that stage, you're really judging it almost attention span.
Yeah, for example, it happened with an article when we have like two themes. First was the antagonistic capability of fungi to controlling pests, and the ability of those fungi to resist to pesticides.
Okay.
So the story was good, but we didn't have like plant assays. So every journal we send it said, no, it is a good story, but we need like plant assays. So okay, let's put it another way. Let's talk with other people and see what they think. So what happened is that we split the work in two stories. First, the story is the antagonistic part, and the second story is only related only to the fungicide resilience. So that's how it is. So you need to change everything, and those changes take a while because you need to rewrite things and maybe do some extra experiments.
Well, when you're at that sort of deliberating on what you're going to do, especially at that stage, once all of the data is collected, how does breaking that up make you reflect on the hypothesis that you had, let's say, a year prior, a year earlier, to having those results? Does it make you feel like you needed to change what your... I suppose I wonder if you're thinking in hindsight, if the hypothesis was different, might the data collection have been improved or?
Yeah, definitely. I think that that the more you do the things, the more you realize that the first thing you did needed some improvement. Yeah, yeah. For example, with the collection. For example, it happened to me with the coding. So I used to have like a large code. Useless. So you need a short code, precise code, easy. So.
Gotcha. Awesome. All right, let's break into Jeffrey. So same questions. Like so. Does first of all, does that sort of puzzle analogy work okay with you?
Yeah.
I mean, I think about it more like a diameter. So, like a circle. So, when you're first learning, like in grade school, you expand the circle very fast. So you learn about mammals, you learn about literature, you learn about all these things, and it expands and it expands, and then you get to undergrad and you pick a major, and it narrows to like, okay, we're just going to learn about math. Okay? And then you get to graduate school and it really narrows further, and you say, I'm just going to learn about endophytes. And that seems like really strange when you think about it, but actually that's how the circle widens. You have to sort of push a very little tiny point out, and then everything else can follow, and then a little bit more, and then follow. And so what I think about less than a puzzle piece is more like, where can I push at the edge of the circle to make it, to widen it.
No, I really like that. That's a cool way to think about it.
It's not my idea. There's a visual of it online somewhere that I read and it made sense to me. But you're just kind of this tiny little point right at the edge of the circle, trying to widen it just a little bit. And it may not seem like a lot, but if over time that circle gets wider and wider.
So it almost sounds like at the periphery of maybe not your fungi understanding, but you might have had questions in your other attributes of life, and that learning that little bit has answered a portion of those other questions. Is that a fair way to put it as well, or?
Yeah. Or I think both academic questions and personal questions. So, like, for example, a big question everybody asks themselves is, how do I want to spend my time?
Yeah.
Right? And if you realize, oh, I really like doing experiments and I really like working with fungi, then, and you spend your time in this one area, and it answers that question.
Yeah.
Okay, I like doing this. I can do this for a while. I think that's a good way to think about it. I'm a little bit more practical in how I think about graduate school and I think than some, but I sort of view it as a way to think about interesting things.
Yep.
And I don't think I'm less inclined to think about it as like I'm doing some really, really lofty work to solve giant problems, although that is certainly what I'm doing. But I think if you think that way, at least if I think that way, I get sort of depressed because it's so hard, and the questions you're asking are very small and they make a difference. But I like to think about it. It's easier for me to think about it in terms of like, okay, I have a set amount of hours in a day. How do I want to spend them? And what will this do for the world, I guess.
Yeah. Okay. Awesome. And so let's lead on from where that took you to the hypothesis you're working on right now.
So right now I'm in a very different spot than Efrain in terms of where I'm at. So we're just now starting to collect data, and basically the project that we're working on is seeing spillover of pathogens and endophytes and fungi in general from coffee fields to forests. So we sample the plant community in the forests and culture and analyze what endophytes are growing, and pathogens are there, and you can compare that across species, you can compare that across distance, and when you make those comparisons, you can say, okay, maybe we're seeing a lot of disease right near the edge compared to 100 meters in, and so, or maybe we're seeing a lot 10 meters in, and so when thinking about forest fragments, there might be a high pathogen load or endophyte load closer to the edge. So that when forming the hypothesis, we were thinking about, what kind of questions can we ask, and what hypotheses do we want to actually test, and I think we kind of settled on looking and comparing. So if coffee diseases are present in high quantities in coffee fields, as you would expect, then plants that are more closely related to coffee will share more of those pathogens and endophytes to a certain extent. So so if that's the case, then we will expect to see higher rates of coffee-associated diseases near the forest edge than further in.
Okay, gotcha.
Yeah.
Okay. How do edge effects normally work with pathogens? What did your, I guess, pre-research reveal about that?
Yeah, so there's not a lot of work done on fungal pathogens moving between agricultural land and forest, but the work that has been done in where I'm from, in the temperate region, always tends to focus on what is moving from the forest into the field. So a lot of times people will think of the forest or the landscape as a reservoir for disease that can then come to your crops.
Right.
But if you reverse that and you look at the other direction, you have a ton of one plant all in one space with very specific conditions. It's going to have really high rates of disease, and that's going to spill over in the other direction too. So when doing reading different studies, a lot of times you do see studies that look at sort of what's coming into the coffee field from the forest. We wanted to sort of pursue the question in the other direction, because that's an area where not a lot of work has been done. So
Okay.
yeah.
So I don't think we've touched on this yet, but what makes coffee vulnerable? So you're saying, like, in that sense, you're saying, I guess in theory, like, you've got a small population, and then if a disease comes in, there's a low immune system to that. Is that what?
Yeah, so I mean, if you think about it from an ecological perspective, if you have a community, let's say, like a community that's really complex, lots of different species. If you have, say, one coffee plant among hundreds of other species, and that coffee plant gets sick, and there's another coffee plant maybe over here, this coffee plant is insulated from this one, from this disease, and so it can't. It still can move over there, but there's all kinds of other things happening around it, but when you pack them really close together, let's say you have one rust propagule that lands and it starts to grow, it has, everywhere it goes, it has a suitable host, and so it exacerbates and gets really, really intense. Yeah.
All right. Yeah. Well, so how have you gone about your methodology and creating that? Well, I guess what did you state your hypothesis explicitly, and just what variables were within that?
Yeah. So if I guess our hypothesis is that we expect to see higher rates of coffee-associated diseases among coffee-related plants near the forest edge when compared to further into the forest. And the way that we've gone about sort of structuring a study is, you have to sample plants at different distances, and you want to sample plants of, you want to sample enough individuals to get a robust picture of what's happening, because if you just sample one, that's not really enough to say anything about it. But you also want to sample enough at each distance, because if there's, effects of distance, you want to be able to compare the distances. And then you also want to sample plants that are different distances phylogenetically from one another, because the hypothesis is that more closely related plants are more likely to share pathogens. And so if you have a very, very distantly related plant to coffee, and you look and you see, okay, the things that are living on this leaf are really, really different than this, then you can maybe say, okay, coffee-associated diseases are really intense on coffee-related plants. And so, yeah.
Awesome. And so, like, you're more or less just starting the data collection.
Yep.
Yeah. What has surprised you? What, in terms of what you expected? What has surprised you? What was as you expected?
I think what has surprised me the most is just the sheer amount of time that each site requires. So I think I assumed I was naive coming into this.
Yeah.
I always am, and you don't know until you get to a place and try. But I think thinking about the amount of work that I have, it's in the amount of time I have to do it, it's like shocking. It's shocking, and so I think there are things that we will have to adjust, certainly.
Okay.
But so that's been really surprising. Things that have been nice have been like, oh, we do tend to see plants. I was thinking maybe the forest would be so diverse that you wouldn't find the same plant.
Yeah.
You wouldn't twice,
Right.
and it would be impossible to find individuals. But we've been to one site, and it was pretty successful in being able to find 5 individuals of each species at each distance. And I thought that was pretty smooth compared to what it could have been. I could have been there for a whole day and not had any.
So you're losing some time in processing, but you're gaining some inconvenience of, like, the actual site itself.
Yeah, maybe not gaining some, but maybe just preventing more loss.
Okay, gotcha.
But yeah, I think, just as I was sitting here, I was thinking about sort of the timeline, and I don't know when I'm gonna sleep. But I think that those kinds of things happen. I've never spoken to a researcher who hasn't had that problem and had to adjust. Like Efrain was saying, you have to be flexible, and things that you plan that in theory are really, really well designed are completely non-tractable. You can't, non-starters. So I think that, yeah, there's going to be, at least in my work now that we're starting the data collection, one of the biggest questions we're going to have to answer is not necessarily what is the data saying, but how do we collect it in a way that is possible in the amount of time that I have.
Yeah.
Yeah.
So it's probably definitely jumping the gun, but have you thought about what I was asking Efrain, the depiction of the results? Is that something that's in your mind?
It
Or
was
at least sort of
yeah.
conceptualized? Yeah. What, I know it sounds like it's something that's going to change over time, but what was in your mind?
Yeah, so basically when thinking about communities, what's difficult is if you have just a list of names on a table, that's not very, it's not easy to visualize how similar those communities are. So you want to think about ways to visualize communities so that you can say these two communities are more similar than these two communities.
Yeah.
And my experience with data representation is mainly just how do other people represent data?
Mhm.
Yeah, how are they making their figures? And the figures that are really elegant are ones that can visually show you a comparison that is very abstract when written. So, like, okay, we're seeing a lot of this and not a lot of this in these two different sites. How do you show that in a way that is compelling and like aids the reader?
Right.
Yeah.
Okay. Yeah, it's really interesting to hear you guys discuss that you're you're actually thinking about the reader, and you think it does sound like you're thinking about attention span, and I suppose, well, making it digestible,
Mm-hmm.
and I mean that's the center of that question. Um, I think. Well, I'm looking forward to hearing more about where do you guys take this, but do you want to, we can start the final questions, and
Yeah.
it'd be cool just to hear about you guys discuss these, and
Mm-hmm.
I'm not sure if you discussed them previously, but yeah. So we already have mentioned them. That said, as I said, think of these more generally, but provide your own spin on them if you have one. So in terms of motivation and drive, or curiosity, or whether or not you're outcome oriented or not, how have you found it as the best way to approach research? And I suppose these are something that comes into the factor of that initial search for information for something to study, but also I think it's also within each choice you make. So yeah, what would you describe is your key motivator to do research, but also this specific research?
I think for me it's a way of finding a fun way to use my time. I spent some time working out after school and found that to be really not engaging, and I struggled with things like, just personally mental health things. I was just like, this is not working. I need to do something else. And I think asking questions and figuring out ways to work on them, and you get to be outside, you get to travel, you get to learn really cool techniques and keep learning. That's really fun, and so that was like the initial motivation to keep going. But as I do it more, I think you have to have kind of a as even as much as you may love fungi, it's going to be hard a lot of times. It's going to be work. It's going to be a job, and so I think to as I've kept going, I sort of am thinking about it more and more as like a way to express myself, because if I don't do that, then I'm just like a machine. So you want to do it in a way that is balanced, I guess. And so I'm still figuring that out. You know, how do I manage it and maintain like my humanity? um, so yeah, I don't know if that answers your question or.
No, absolutely it does. Yeah. What about you, Efrain?
Yeah, for me, it has been like after finishing my bachelor in biology, it was like okay, I immediately have the contact for the masters. So my thought was like, yeah, why not? It looks like amazing. I like to be outside. I like to study those fungi. So yeah, it's like being in the wave.
Yeah.
So as soon as I enter the masters, yeah, everything gets more complex, but also your desire for discovery grows too. So you keep going, and then because of relations of your advisors with other advisors around the world, you start to know more people. So you don't know when you are already connected
Right.
before you
Yeah.
start, because for now I'm here with them.
Yeah.
So uh, I I wasn't expecting that for this year, for example. No, I was just expecting for this year, hopefully to finish the PhD and maybe starting to work as a researcher or something like that. But now it's like a window is opening for doing something else.
Yeah.
And what it's gonna be, I don't know. But
Okay.
yeah, it's amazing.
Yeah.
So like for I think I'm fortunate to to be able to choose every every single way on my life, because yeah I feel very free to be here, for example,
Right.
instead of staying in San Jose, or never being here in Costa Rica and staying in Mexico. So yeah, I think it's been a lot of hard work, but also I don't know, sometimes it's just luck maybe, or
Yeah.
circumstances, random circumstances.
Yeah.
Yeah. So it's
So
interesting.
I wonder, as things have become more complex for both of you in understanding, has it been, I suppose, well, it sounds like the more people you know, obviously, the more you're learning, but also, is there a push factor there that you see you're surrounded by people who are producing a certain standard? So I'm trying to explore whether or not it's like a healthy competition, or if it's like or it's just you know, you on a daily basis you're seeing other people's work ethic, and I'm wondering whether that shifts that drive.
Yeah.
I mean, so we had a very interesting conversation in one of my classes where we were talking about sort of scientists' workload, and we were reading a book by a very, very famous scientist, E.O. Wilson, describing, you know, what being a scientist is like and what kind of commitment you need to have. And he described sort of like you have to be, you know, constantly, you know, working, and you don't take vacations, you take field trips. And a lot of us were really uncomfortable with that in the class, because it's like there's a reason why the suicide rate is high among graduate students compared to the overall population. And I think for me, one of the challenges is, as I get more into it and more interested and more motivated, and it becomes very easy to become really obsessed and completely unhappy.
Right.
And some people can do that. You know, like my brother's an example of someone who's a researcher who just loves to. He would never do anything else. He just
Yeah.
loves it. But for me, like, I have other interests and other avenues that I want to explore, and so I'm navigating that. You know, like, how do I? And I think there maybe is a generational divide. So like older scientists who are they grew up in the school of like you really need to work really, really hard and pursue your career, and younger people, my peers at NDSU especially, are thinking more about like, okay, how do we balance ambition and quality of work with also like, maybe we want to raise a family, maybe we want to, have a home, and so I'm figuring that out. I don't know the answer, but I know that like what we do requires a lot of work, a lot of time, and you don't make $1 billion, and so like figuring that out has been a challenge, and that's really where I've had to do the most navigating as I've gone through it. Like the learning part is, I know how to do that. I know how to like read and try things and experiment. But what I've never experienced before is like devoting all of yourself to one thing, and what that can do to your your health.
Right. Yeah.
Mm-hmm.
That's I didn't expect that answer. But that's really really in depth. I appreciate that. Do you have a similar thoughts, Efrain, to that? What do you think about that?
Well, I think that maybe the grad school can be like different accordingly in where you are. For example, I think in the US, I don't know, it's like maybe, how can I describe it? Like, it seems like a cold environment. So, for example, and here in Costa Rica, people, well, and this wasn't happening also in Mexico. This is only like a thing from the Central America, maybe Colombia. I met some people from there. Yeah, so for example, we have here like coffee afternoons. So every single day of the week, at between 3 p.m. and 4 p.m., everything stops and everyone is on the dining room to take coffee. So you start speaking about everything, ideas, troubles, academic things, personal things, whatever. And it is a big release of stress, and you can get answers that you weren't expecting. So it is nice, and I think that this is not happening in the US.
Yeah.
Hmm. In the US, I think it's an article of, I think was Science, about the stress that is put upon grad students in the US, and they say that yes, it's like a very solitary pathway. You are on your own problems, on your own issues. So it's difficult to have a nice talk with
Yeah.
someone else. And this is not happening here in Central America. In Mexico, it was, because after coming here to Costa Rica for 3 months during the masters, I realized that it was really nice to have a break during the day in the lab and start talking with people.
Right.
When I came back to Mexico, I tried to do it, and it worked. It was like, yeah, it feels nice.
Yeah.
And well, sometimes it was not coffee. Sometimes it was a beer. So it was cool. Also, so yeah, I think these social things, relations, are strongly needed to keep the mental health, and also to be like better persons, because maybe you have like you share a hobby with one of your colleagues, and you don't know until you spoke with each other. So in that way, you can have all the spheres of your life like in this thin balance, like the family, friends, academic things, and stuff like that. So here, I think for me, as a PhD student, it has been like a very challenging in the part of the academic, but also in the sphere of social and personal development. It has been very, very nice. I mean, I haven't feel alone, despite for not being here from Costa Rica. So yeah, it's nice.
Okay. So my next question is about risk, and to me, it sounds like both of you are factoring in personal health into that, and which is really interesting because, as I've sort of explained to both of you, like I've been looking at decision making heuristics, and there was like, you take alternatives, you apply markers of progress, and you have that cost to acquiring information. And that cost is also sounding like health, right? So it is, I say to people, I say it's time and energy, but those really, they funnel down. That's the generalized factor. And if you're talking about money, money is representative of time and energy, which is, of course, framed individually. So, I have these questions about risk associated to hypothesis and method, but it also just really sounds like the fundamental factor is, where am I going? Whereas what is my overall aim? Not just the aim of my lab, but also the aim of you as an individual,
Right.
or your family, or where you're from. So it's actually really touching, kind of, to hear both of you discuss that, because it's not an answer I expected. Would you think there's any other risks that we haven't mentioned that kind of come into your mind?
Of course, there's the risk of employment. So you invest time and energy, and you maybe sacrifice years that you could have started a family, or you could have bought a home, and then you get your PhD, and there's no jobs.
Yeah.
And so that's always a risk, a very practical risk, but one that is real. And I think that's why it's important to, I think, and there's also the risk of maybe your work in many ways doesn't do what you expected it to do. You can't get it published where you thought you were going to get it published. And so if you tie up a lot of your self-worth into the work, and it feels really good when you get an award, or you get published in a high-impact journal, but it feels horrible if, say, your data is no good, or your methodology gets ripped apart by a reviewer. That can feel like you're getting ripped apart by a reviewer. And so I think one of the bigger risks I see going forward is getting really tied to my worth, tying my worth with my work. Because if you do that, it's really hard not to do that because we culturally I've been trained to view work as what I am worth. So that's a pretty big risk, because let's say you get out of graduate school and all of a sudden you don't end up in a position where you thought you were. You maybe don't have the job you thought you had, or you take a different path, and maybe that could be devastating to you, or it could. But if you have the mindset of like Efrain said, like, you never know what happens, what door opens. If you view life as that, if you view like your career as that way, then it becomes easier to deal with those things, because they aren't disruptions of a path, they're just a different door. I think one of the hardest parts of graduate school for me has been, my whole life has been basically, I don't like to plan. I like to allow things to happen on their own and be surprised. But graduate school is like, you have to have a plan, you have to have a research plan, a budget, you have to think not only about your master's, but maybe, do you want to do a PhD? Where do you want to go? Where do you want to live? What kind of jobs do you want to have? And that gets you thinking 10 years down the road, and I'm not used to that at all.
Right.
So that's been an adjustment, and I think a risk of that is to never be present, to always be thinking about what is it going to be like in 10 years. So.
Yeah. So that breaks into, I know these questions kind of, it almost feels impossible to do chronologically at times, but it breaks into expectations and adaptability. And so how do you think those two things factor in to what you've just said there? And I kind of have an elaboration on expectations as well in the research process, to say that once you get to the method, you trust the process you've designed. So I suppose I wonder how that affects the research process, but also how it affects expectations more broadly, and whether or not you have to be adaptable in your career as well, because it's sounding that way from what I'm hearing.
I think so. I think that adaptability is important in all aspects of research, but so from the methodology on up to the career path you take, it's really nice to think about, like, if you set your mind to something, you can achieve it, and like to have this goal and to go at it, doggedly, and
Yeah.
you're going to get there. But in many ways, that's not how the world works. And like, I don't trust the process with methodology because I know I will make mistakes.
Right.
I know that it might not be realistic and I'm going to have to adjust. So I think I trust small results in the moment. So, okay, this is going to take 2 minutes per plate, per thing. That's manageable. This is going to take 10 minutes. That's not. So we can't do that. Scrap that. We have to do this. I can trust that. If I have a plan laid out, I don't think I can trust that until I'm doing it. But yeah.
Yeah, because I suppose when you're adding up plates over a lifetime, I suppose if it's adding up the plates on a daily basis and you're thinking in that way.
I sort of say that because that's all I've been thinking about the past year. But yeah.
Is this my life forever?
Yeah. But you in my case, part of the reason why this is so intense right now is because I'm not here for very long. If I were here for 5 months, it would be a different story. I could take more time. But yeah, I mean, I'm sort of obsessed with that right now just because that's at the level I'm at. I don't really think about data representation too much right now. I don't really think about statistics and analysis. I'm more just thinking, what's the method that's going to be working?
Right.
And I think that's just where I'm at.
Yeah.
When I get further along, then I'll be obsessed with graphs.
Right.
Yeah.
Yeah, no, that makes a lot of sense to me. What do you think of Efrain?
Yeah, well, I'm at a different point because I've been through the process of processing plates, yeah. And now, well, I need to work with mostly on the computer with data sets, writing stuff, but also meetings with advisors, meeting labs for maybe for if I'm invited to give a talk about a specific topic of fungi. For example, my results with the bioinformatic stuff or maybe the chemical stuff. So your agenda just keep growing on the way. So when my advisor says that you thought that agenda will be shorter once the PhD is done, and it's not, it gets only bigger. Yeah, because now, for example, in my case, this week I'm here in Las Cruces. Next week I need to be in Los Santos in a greenhouse working with my coffee plants, but also I need to be on Zoom meetings with my colleagues and guys from the other lab, which is the Sandy Biot in San Jose. And eventually, all the Fridays, I need to make a room for a weekly discussion with another professor. So it's like you need to. I think the PhD helps you to manage your time because sometimes you can be like very full of things to do, but you need to remind yourself that you're a person. So you need to eat, you need to sleep. If you like to do exercise, you need to a space for doing exercise to release the stress. And always, it's good to have like a nice meal. For example, going to a restaurant to have a couple of beers, and okay, you need social life too. So you need to make room for everything in your life. And what is interesting is that it doesn't matter what are you doing, and you keep thinking about work. For example, when I'm biking, suddenly I'm like, oh, maybe that can work, and then I go back to home, and yeah, it works. Sometimes it don't. Yeah, the ideas never stop coming. So yeah, it's I think being a grad student, master's, PhD becomes a lifestyle, and it's an interesting lifestyle. I mean, it's not for everyone, but if you are like comfortable with it, you feel happy. Yeah, it is for you. So I've seen all kinds of personalities on my advisors. Ones that are more like only work. They don't have a family, so it's a big sacrifice to be on that way, because uh, they have sacrificed not to have like family parties and stuff like that, or maybe not having the, the amount of holidays that all the professors have, maybe. And you have the other part, people that uh, know how to manage their time, to have family, to work, to attend the students. So everything is possible, and which way are you going to choose depends on what, in which way you are comfortable with. But challenges never stop, and yeah, that's that's how I see it so far.
Right.
Hmm.
Slightly different train of thought, but how do you guys see cooperation within research or within the stage you're at? Because you were getting at the idea that it's very individualistic in some places.
No, it's fundamental.
Yeah.
Hmm. Yeah, I think, for example, now for the time I'm here with them, I, I know that we are going to collaborate eventually in a paper, which is how we measure the things in science. So, yeah, I think the cooperation is it's a fundamental piece for doing big and good research, and also for gaining funds, maybe international funds even. So, yeah, and the basis of cooperation is relationship, to be like be kind, and yeah, just keep working, and the good work will spell will speak for you. So yeah, I think that's the key. Hmm.
Okay. Because that sort of breaks into, if it might be a fair assumption to say that if things are more cooperative and everyone's working well together, that will be a pretty satisfying part of the process. But I have a sort of separate question whether about satisfaction or dissatisfaction in the process as a whole. So would your favorite part about this all be cooperation, or maybe something else?
I've been in the both positions, when the cooperation works and when it doesn't, or it doesn't work at the full. Um, but that's life, so you need to try. If you don't try, you will never know. So at the end, it depends on the effort you put on the task and how you shape the results. So maybe you were expecting a big thing, but the cooperation doesn't result like you were expecting. But I'm still working for doing something smaller, but it is work, so you need to finish it. So, and in the other case, we have been involved in where one idea was like a small idea, and more people comes in, and the idea grows bigger, and the work is like, wow, it is nice. It feels like good. And everything is running smooth. Yeah. So.
Your thoughts there?
Yeah, I mean, I think I agree with a lot of what Efrain said. I think one area that um, is also important is cooperation more broadly. So, like, friendships outside of your field, um, things you do elsewhere, family relationships, all those things. Like, um, yeah, science is cooperative in the sense that, like, it's fundamental. You have to be able to work with others. You have to be able to share skills and to some extent, know weaknesses. But also, you need to be able to cooperate um, with other people. Like, I think about how um, you could have a conversation. So I come from a family life of a not very educated family in a not very educated place, and so for example, when I talk about what I do, like methodology and theory and all these things, it just, like, it's a non-starter for conversation. And that's not how you keep relationships alive. And so, for me, this is kind of answering a different question, but like, you have to be able to know when to, and how to communicate about what you're doing to people who may not care or may not know what you're talking about. And, that's true of any profession anyway, like
Right.
it's not just true of science, but like you have to be able to shut it off.
Yep.
and talk about other things, sports,
Yeah.
yeah.
No, but you've made a segue, but it's the segue makes sense, because you've touched on my two last questions, and it's the idea of responsibility and science communication. And so you're saying, it sounds to me as though you're saying, in that responsibility of communicating your own work, that you need to, there's times when you need to choose your audience. So,
And you also need to choose when is it important to share. So, like, I it's not like when is it important that I talk about statistics with, that's a very strange example, but like you want to be able to, I think I want to be a person who has the ability to interact with all kinds of people. And I think that's important and healthy. And so part of the balance of science communication is choosing your audience, but knowing like when to be an audience. So when to listen to people, when to ask them questions, and when to learn about other people's desires and dreams, and what they're doing with their lives and what their jobs are, and what they think about. And so, yeah, I love communicating science, but I also think we are also responsible for being well-rounded.
Right.
So that takes practice too, like anything.
Okay. What are your thoughts there, Efrain?
Yeah. For example, I think I'm fortunate to be surrounded by nice professors in the university, and they are also like very good people, because one of them always says that I'm not an expert on what I'm doing. I'm here, and I'm good because I have found the right persons for some tasks. So I think that's one of the best advices, because yeah, you need to be able to cooperate with another scientist from another field, for example physics, mathematics, and then a question just get bigger. So also they also talk about how they live. For example, if they have had bad experiences in life, they tell you those stories so you don't make those mistakes. So it is nice to have those advisors on near to you. So it's really nice.
Awesome. Cool. Well, I think we're at 1 hour 40. That's pretty
Wow.
good. That's
Yeah.
pretty good, guys.
Yeah.
Yeah, I really appreciate it. This was like a really enriching conversation.
Yeah.
I appreciate it a lot.
Likewise for me as well.
Awesome. Well, we'll 100% follow up on where you guys are at. I'll just keep posted, I guess.
Yeah.
Awesome. Thanks, guys.
Portent Tide is independent from the researchers on the podcast. If you wish to support them in any manner, a link is provided from the researcher to allow for this. This can be found on Portent Tide YouTube channel, Instagram, and directly through the Portent Tide website. If you enjoyed the podcast and you wish to support Portent Tide produce more conversations like this that provide an insight into how researchers work, you can do so directly through the links provided on the Portent Tide website or through wherever you found this podcast. Thank you again for listening, and any support and feedback is much appreciated.